I'm going to talk today about population geroscience. I know all of you know the first word in that title. Can I see a show of hands for how many people have encountered the second word? Okay, that's what I expected. So good. I'm going to explain what this means. And actually, I'm going to explain what that means almost immediately. These are my disclosures. In particular, there's a piece of intellectual property that I'll spend a lot of time talking about. It's being sold to consumers. It generates regular revenue, but I do receive a royalty check, so I disclose it. And it's freely available to search. This is the substance of the talk. I'm going to explain to you what this geroscience thing is. I'm going to talk about the work that we've done in that space to develop measurements of this thing we call biological aging. I'll conclude with some examples of how we've deployed these measurements in the context of population science, and then I'll talk about what we're up to next. And if I make it all the way to the end, I will even tell you about a resource that you can access at Columbia if you're interested. Okay, so there are many social scientists in the room, I presume, and in the social sciences, aging is studied as lots of different things, including a process of personal growth, actualization, we call it adult development sometimes. And I want to just be very clear that as I talk about aging in the biological space, it's not meant to take anything away from that notion of aging as an opportunity for generativity, for becoming something. But as we study aging in the biological sciences, it's primarily a process of unbecoming or deterioration. And the working definition that we use is this progressive and accelerating loss of system integrity and resilience capacity at the cellular, tissue, and organ level with the passage of time. And at the bottom of the slide are two cartoons that I think are sort of useful for summarizing the state of understanding of this phenomena. Let me start with this one. It's sort of a better graphic design. This was a picture that was first published in 2013. I think this is the 2023 iteration. But it's outlining a series of molecular changes that characterize the cell, in particular in mammals, but these have been observed in other animals as well, as they grow older. And this conserved set of molecular processes is mechanistically linked with the deterioration in the functional capacity of animals as they age, as well as their risk for the animal versions of the many chronic diseases of aging that we invest all of our, well, maybe not so much anymore, but historically anyway, billions of dollars in studying in health research. They include things like telomere attrition, which may be relatively familiar. That's sort of like an old school biomarker of aging these days. It was a hot thing when I was a PhD student. It's since sort of fallen out of favor. At the cellular level, it remains a fantastically important biomarker of the aging process, but at the organismal level, it's much harder to interpret. Loss of proteostasis. So, you've heard talks about genetics from Dalton, Sam, and others, no doubt. A thing that genes do is they encode proteins. The proteins are sort of amino acid origami. They're these exquisitely folded molecular structures. And as we get older, we get less good at that folding. So we make more errors. And we get less good at identifying the misfolded proteins and chopping them up and reusing them for spare parts. So you get misfolded proteins floating around, causing trouble in cellular systems. Cellular senescence is something else you may have heard of. It's what happens when telomeres get too short. It can be caused by other things as well. And it's not quite death. It's like a zombie state in which the cell stops doing its job and starts floating around and emitting pro-inflammatory molecules, kind of recruiting other cells to come in and kill stuff, which can be bad news. Epigenetic alterations, which I'll talk about a little later, and many others. So the point here is that we sort of know, at least to a first approximation, the molecular basis of this time-dependent deterioration in the biological integrity of an organism. And that has motivated interest in this concept of geroscience. And sorry, I realize I've neglected Luigi's figure here. Luigi is pointing out that aging is essentially the interplay between resilience processes that try to keep our biology together and the sort of entropic force of time. and eventually time lengths. So these cellular hallmarks accumulate despite our genome's best intentions at maintaining the integrity of our biology, and eventually we die. But because we have characterized these molecular processes in this way, scientists have proposed that they might provide a novel opportunity to intervene to prevent the deterioration of organs, the onset of disease, and to preserve healthy lifespan. So this is my sort of geroscience cartoon. Aging begins with this accumulation of molecular changes within the cell. It progresses toward a loss of the integrity and resilience capacity of tissues and organs, which in turn impair our function, make us sick, and kill us. And by intervening, either chemically or behaviorally, or as I'll suggest perhaps environmentally, to slow the accumulation of these molecular changes, we can delay or prevent the loss of system integrity and preserve health span longer for more people. That's the promise of geroscience. Now, geroscientists, as a general rule, who come out of the field of aging biology and in some cases clinical medicine, think of aging as an intrinsic process. This is something that is a natural consequence of time. The processes of living are the thing that ages us. We have to metabolize nutrients, for example. Doing so generates molecular damage. When we do all kinds of things in our lives, we experience molecular damage. Radiation from the outside, the imperfections in the air we breathe and the water we drink. All of these things are understood in aging biology and in most geroscience as the inevitable consequences of being alive. And so what they're interested in studying is the sort of natural unfolding of this process, genetic differences between organisms that may make them more or less resilient to it. But of course, everybody in this room is familiar with another set of processes which may drive differences between individuals in the rate of accumulation of molecular damage and the progressive accumulation of these hallmarks of aging, which ultimately cause disease, disability, and death. So I don't sort of need to belabor the point with you, but this is just to say that it's not just being alive that ages us, it's where and how we live. And again, this is a slide I use when I'm talking to the sort of classical geroscience crowd about how differences in environment and behavior can actually cause differences in aging. But essentially what we're talking about a direct path through which toxicants in the environment can enter our bodies and interfere with our cells, and an indirect path through which the transduction of social environment through, for example, stress biology or the regulation of behavior result in that kind of damage. Again, this is old hat for you guys. But just to say that all this biology is very deeply connected to the environments that you spend your lives studying. So what are we doing? This is a really easy process to study if your focus is the laboratory mouse or C. elegans, or if you're really lucky, yeast, because those things age fast. Mice live a couple of years, worms, months, yeast, days. So lifespan, or in a mouse now, signs of health span, so reduction in mobility, for example, are things that are pretty easy to study longitudinally within the course of a 5-year NIH grant, or certainly a scientist's career, or even a postdoc. But humans live an awfully long time, and that presents some important challenges, and that's where this work on measuring biological aging comes from. So we can't sort of wait around for people to get sick in order to study their aging process. So in this cartoon, I've got time on the x-axis, beginning here in midlife, let's say, heading into later life, and then the y-axis is this underlying biological process of aging I've been talking about. And the color coding of the slide is meant to indicate what your doctor sees when you go in for your annual physical. There's sort of like a period of life when you look like you're healthy, and a period of life when you look like you're sick. And what is illustrated then, here with the colored lines is a person who experiences an elongated health span in blue, and then someone with a shorter health span in red. And you can see they look different to the clinician because they are different ages when they cross over from healthy into sick. But what I'm highlighting in the figure is that there's this long period of latency in which these trajectories of biological aging are diverging in advance of the onset of disease. And if we could ascertain this, then we might be able to intervene to prevent. Now, right, so we would want to intervene to prevent way back here before the trajectories diverge. But of course, the follow-up time is prohibitively long, both from cost and time considerations, to actually run a clinical trial that tests whether treating somebody today will keep them from getting sick 20 years. So we attempt to measure this process. Now, it used to be the case that we did this in, I guess, what I would call a hypothesis-driven fashion. And this is where telomeres came from. They were known to be mechanistically important to the life of the cell. And so we measured them in people's blood to see if they could do the same thing at the organism level. You also have sort of various types of frailty indices, something like allostatic load, where we sort of engineer the measurement from what we already know about the function of biology. And these days, we are moving in a different direction toward a data-driven approach to the measurement of underlying biology. And so the way the biological aging measures I'm going to talk to you about today were developed is by assembling comprehensive data on people, either through blood chemistries and functional tests and scans, or through omics. And you no doubt can't read this in the back, but it says genomics, epigenomics, transcriptomics, proteomics, metabolomics. There are probably other omics that I have not mentioned because Thermo Fisher didn't put them on their little icon. But there are all these ways of comprehensively assaying the molecular structures circulating in our blood or apparent in other tissues. And we can then use machine learning, data mining, I guess now it's all called AI, to extract from these large data sets signatures of the aging process. And we'll get to like what the label we need to use for that machine learning is. But the theory, and in fact, now we have about a decade of evidence on this to support it, is that this approach will give us measurements that are more variable in younger people than the indices we have historically used to study individual differences in aging. They will allow us to ascertain differences in risk before disease's onset, and ultimately, they will prove sensitive to preventive intervention. And that last one is probably the piece we have the least evidence on, and this is the most important, but I will speak to that by the end. Okay, so I'm going to start with where I started. So if you were here 9 years ago, you already saw this slide. I think I was presenting from the other side of this room. Nice. Okay, so I do remember, it was not a good day. It was election day, 2016. Well, yeah, for me it was. You guys were loved. Today it's not starting out great either. Now, there might be something here you might reconsider future invitations. In any case, we were interested in geroscience theory and its translation into human populations. And it seemed like the purest test of that was to see if we could measure aging in young people, people who were not conventionally understudied in gerontology. And we had this unique opportunity because of the incredible work that had been done by my mentors and colleagues, Terry Moffat and Avshalom Caspi, in the Dunedin longitudinal study. So this is a unique study of a complete birth cohort born between 1972 and 1973 in Dunedin, New Zealand. And they've been followed up now for a half century. So phase 52 is in the field now. They're on track to retain 95% of the surviving birth cohort members. They've done that now across, I don't know what it is, 15 assessment waves. But importantly for us, beginning when the study participants were 26 years old, they were brought into the laboratory at the University of Otago, and they got a complete physical workup. So there were blood chemistries done, cardiorespiratory fitness testing, lung function testing, a dental exam, the works. And those same measurements were repeated again when they were 32 and again when they were 38. And so we were able to align across these three assessments 18 measurements of the functioning of different organs in the participants' bodies and model changes over time. So the figure in the middle is showing you that if we score things so that higher means older, based on what we knew in the literature of what the age curves of these biomarkers look like, we see that even in these young people, these biomarkers are moving in that age-related direction. So that was important, particularly for convincing gerontologists that they should care about aging in young people. But we were particularly interested in the individual differences. And so we modeled change in these biomarkers using mixed effects growth models. We preserved the random slope terms from those models and combined them to form an index that we called the pace of aging. We scaled it by the mean in the cohort, so that a unit of 1 corresponds to 1 year of biological change per calendar year. And then, it's a ratio of that. So if your pace of aging is 1.5, it means you're aging a year and a half for 12 months, or 50% faster, and the reverse if your pace of aging is 0.5. So this was important, but it was limited in that we had generated this measurement of aging, the aging rate, in this cohort, but it was virtually impossible to export, although not completely impossible, as I'll show you eventually. Meanwhile, something else was happening. So this is a cartoon picture of DNA methylation, and as we were phenotyping the aging rate from repeated measures of people's blood chemistry and organ function, Steve Horvath was changing the world by revealing that from essentially any cell in the body, he could extract DNA, assay chemical modifications to that DNA called methylation marks, and from those perform simple machine learning analysis to derive an algorithm that could tell him when the organism was born. Now, in the beginning, the organism was just a pus. But he assembled data that had been generated by other researchers and posted on the internet, spanning fetal development through 100 years of age. And he built this algorithm that could predict how old somebody was in any tissue in the body, which is really cool if you consider that my blood cells are at the long end, maybe 20 days old, but each one of them knows when I was born. So Steve has gone on to show that he can build these epigenetic clocks for essentially any animal you bring him, and he's done other things with it as well. So this really changed the way the aging field operated. And it was proposed that the errors in this prediction algorithm could be interpreted as readouts of biological aging. So this figure that Bernice Benayoun put together in a review article is illustrating the concept. So here's chronological age on the x-axis, biological age on the y-axis. Here's a one-to-one correspondence. And they're showing you heterogeneity and aging in the silhouettes, right? So this person is 70, and Bernice thought that a 70-year-old should walk with a cane, or probably the graphic designer at Nature Reviews Genetics. But in any case, meanwhile, there's another 70-year-old in worse shape. So you can sort of crosswalk this over and see this person is chronologically 70, but biologically 90. They need 2 canes. Whereas this guy, is more like 30, 0 canes. So this was a really attractive idea. And because it was so intuitive the way Steve had built this algorithm, people really wanted to believe it. But it didn't really work. We observed this early on and had a hell of a time publishing our paper. We eventually got it out. Everybody else eventually came around to the realization that these things were fascinating, but they were not predictive of morbidity and mortality. They were not an index of this progressive loss of the integrity and resilience capacity of cells, tissues, and organs that makes us sick and kills us as we get older. And so, to their credit, Steve and his colleagues went back to the drawing board, led by, oh, wait a minute, I'm sorry, I'm getting ahead of my story. The question I want you to ask now is, why didn't it work? And now I'm going to tell you more stuff you already know at greater length than the environmental causation of disease. So this is another guy from Colombia. His name is Abraham Wald. You guys probably, some of you recognize this picture. This is from the Wikipedia entry for something that all of you learn in your stat classes. Does anybody want to shout it out? It's okay. All right. This figure should also be familiar. You guys are demographers after all, right? These are survival curves. What they're showing us here is that if you build an algorithm by comparing young people to old people, you are comparing apples to oranges when it comes to the populations you're sampling them from. Everybody is still alive when they're 20, or at least none of them have died because of aging. That is not true anymore when you sample 70-year-olds. So this is where Abraham comes in. Abraham was a statistician who sort of gave us survival bias. And he was tasked with telling the army where to put more armor on the bombers that were flying missions over Germany. And the data his team were working with looked like this. They showed where the bullet holes were on the planes that were landing in London. And he told them, put the armor where the bullet holes aren't. Why not? Why was that the right place to put the armor?
Those planes made it back to London, even with photos.
That's right. So maybe Steve's epigenetic marks, coding chronological age, were a mixture of the signs of the damage he was looking for, as well as the signs of survivable insults that he wasn't, or even markers of resilience to the depredations of aging. After all, 70-year-olds are by definition successful agers. They've outlived, in the cohort Steve was looking at, roughly half the babies born in the same year as they did. So survival bias likely contaminated Steve's model. Something else you're familiar with are cohort effects, right? So if you, in a cross-sectional sample, your age is also your year at birth. And we know that exposures that embed within the methylome, like environmental toxicants, like pathogens, like various healthcare procedures, vary over that time span. And so what looks like a sign of age in the model could instead be an artifact of the particular period in history when the person was born. So now I can pick the story back up, right? So the first generation of clocks, they didn't work so good. So what did they do? They flipped the model around. Instead of trying to predict how long the subject in their lab had been alive, they decided to predict how long they were going to stay alive. And they were actually trying to solve another problem here, which is that, thanks to the work from folks like you, they had an expectation that aging was not a linear process. And so they wanted to model, as the label in their machine learning, something that followed the same kind of exponential function that mortality risk follows. So they modeled survival from the methylome and came up with the second generation of epigenetic clock. So Morgan's clock was called the pheno-age. Ake Liu, another of Steve's collaborators, had a second iteration called the Grimm age. There are some finer points in how these were developed, and they're not quite the same thing as one another. But what I want you to take away is, instead of asking, time since birth, they were asking time until death. And these clocks proved to be much better at capturing individual differences in health span and lifespan, as well as, in many cases, social determinants of health, especially the Grimm age, more so than the Pheno age. But the logic of their interpretation is the same, right? There's a prediction error around the expected mortality hazard given the chronological age, and that can be translated into a measurement of biological age. But these are also not perfect, right? They summarize total risk of death at the time of measurement of the DNA. And the aging process is a process of change. So it ought to exclude risks that a person is born with, for example, genetic risks, or prenatal risks, or early life insults that have stable consequences in the sense that they alter the apex of development, but not the rate of deterioration from it. So these might be predictive of mortality, but they might not be modified by interventions designed to slow processes of aging. And then, the final bit here is that these clocks are really optimized to summarize a life course accumulation of risk. And if we imagine that clinical trial testing the therapy that we know slows aging in worms, flies, and mice, in humans, it's intervening for just a tiny fraction of that period. And so it leaves open the question of whether the measurement is tuned to be sensitive to the kind of variation you can induce in just a couple of months or years. So clearly this is a setup. And it's a setup for yet a third generation of epigenetic clocks, in this case, based on our rate of change measure. And the idea here is we have a young cohort, so we don't have survival bias. They're all born in the same year. We're not worried about contamination from cohort effects. And the phenotype is defined from longitudinal change over an interval in the adult life course. So we know when the variation in the outcome is arising. And we can, from that, build an epigenetic clock, or as we have tried to encourage people to call it, a speedometer for the aging process. And here's basically the design. We're using a fourth wave of data to model the pace of aging, so it's 20 years of follow-up. We're using DNA collected at the end of that follow-up. So the machine learning is essentially looking backward across this 20-year interval to say, how fast has the body changed over the past 2 decades? And what comes out is this algorithm consisting of 173 CPG sites. Linear combination of those gives us this molecular estimate of your pace of aging. So 20 years of longitudinal measurement distilled into a finger prick blood test at one time point, or that's the idea. Yeah, so I've covered this. I'm going to show you a little bit of validation data, but I have more ground to cover, so trust me, there's a lot more where this came from. So these are figures that are showing comparisons of effect sizes of the Dunedin pace clock, which is gold, our first generation clock, which had a lousy name, sort of like IKEA furniture, POAM. They're initializations, but that was just not a good one. In orange, then the first generation clocks, the age predictors are in the kind of maroon and purple shades, and the second generation clocks, the mortality predictors are in the blue shades. The one on the end is Grimmage. Okay. These are data from the Framingham Heart Study. These are this is about 15 years of follow-up from the time of DNA sampling, and it's clinical ascertainment of incident cardiovascular disease, stroke or transient ischemic attack, and dementia, all cause. And so what you see here is that this speedometer method delivers a marker that is comparably predictive to the sort of best in class, which typically is the Grimmage, for these outcomes. And the same is true when you get to survival. Grimmage is probably a better predictor of survival than Dunedin pace, and it ought to be, because if we think about a model like the intercept generally contains more information in it than the slope about a cumulative risk process like that one. But I just want to caveat that this coefficient is not a good estimate of the Grimmage effect, because this is the data set used to develop the Grimmage. So this is an optimized survival prediction in the same data set, and so but Grimmage is a really good predictor of mortality. Okay, so it does a pretty good job at predicting when bad things are going to happen to people. That's true also if you're interested in disability. These are Jessica Fowles data from the HRS. These are data we published with the paper, again, from the Framingham Heart Study. Again, just illustrating that it's not just morbidity, but actual functional ability to live that is related to these epigenetic clocks. Okay, but the whole reason we built this thing was to see if we could test whether interventions designed to slow processes of aging were working. And I guess I didn't show you this animation of the cartoon before, but that's the theory. So we had an opportunity to do this because of a collaboration with the Calorie trial. Calorie is the first randomized controlled trial of long-term calorie restriction in healthy, non-obese adults. Why would they do such a thing? Well, they're doing it because since the early 20th century, we have known that feeding mice less, but maintaining adequate nutrition makes them live longer. And that finding has been expanded across a range of laboratory organisms. Caloric restriction is our best established geroprotective intervention. And more recent studies show that it does slow the accumulation of aging hallmarks, and that slowed accumulation of aging hallmarks mediates the preservation of lifespan in mice. There are ongoing caloric restriction studies with rhesus monkeys, although we'll see what happens to them with the NIA. But the story to date is that it definitely keeps the monkeys healthy for longer, but it's not clear that it keeps them alive. Anyway, in Calorie, they randomized 220 of these healthy non-obese adults to 25% caloric restriction or regular diet for 2 years. The intervention was not entirely successful. So on average, the control group didn't change their caloric intake, although there was a lot of variability. In the treatment group, they achieved close to the target at 6 months, but then they fell off from there. So by the end of the intervention, the cumulative caloric restriction was about half the prescribed dose. They nevertheless induced substantial and sustained weight loss, as well as, and I don't think I have oh no, I do have the slide, a lot of other salutary health impacts, with the caveat that the pre-specified endpoint in the trial, which was resting metabolic rate, was not durably altered by the intervention. And some of the trialists have argued they didn't have a very good measure of the endpoint, but in any case, I encountered this data for the first time shortly after we published our pace of aging paper, and we did an analysis, which is going to be visited in this software package paper, of the blood chemistries, essentially asking whether the intervention delayed aging as measured from clinical blood analytes. And what you see here are sort of blood chemistry versions of an age predictor, a death predictor, and this thing my colleague Alan Cohen calls homeostatic dysregulation. which Noreen is familiar with. This is a biomarker Mahalanobis distance. So you can look up Mahalanobis distance. You probably don't learn this anymore, but it was a way of diagnosing outliers in regression, for example. And I think, yeah, one of you has actually used this package. So anyway, there's software to implement these algorithms in biomarker data if you have access to this. But the point was, we saw evidence that the intervention was actually moving the needle on aging. And we used this evidence to go to the NIA and say, hey, let us have a look at these epigenetic clocks and some other genomics in this study. So this is what the epigenetic clock data look like from calorie. So the data here are being graphed as change relative to baseline. But we have measurements of DNA from baseline, 12 months, and 24 months of follow-up. And we're tracking change across the interval. And what we see is that the pheno-age and the grim-age clocks, these mortality predictor clocks, they tick. People get older year on year. And that's pretty cool. But there's no difference between the treatment group and the control group in the outcomes. In contrast, for our speedometer measure, what we see is deceleration from baseline to 12 months and then maintenance of that slowed rate through 24 months. There is this sort of hint of an increase in the control group. And I don't have an explanation for that. We can speculate and say it's regression to the mean. You have to be healthy to get into the trial. So maybe people were sort of living their best lives in order to kind of meet the inclusion criteria, and then they revert back to normal lifestyle, and we see some of the slippage. It's not a statistically significant increase, but it is there. And you could argue it's inflating our effect estimate. But we do see this reduction in the rate of aging as measured from the methylome that's maintained. But, you know, extraordinary heterogeneity, which one of our reviewers attributed to genetics, and we may imagine reflects a combination of genetic and life history factors that modify the way people respond to caloric restriction. Reassuringly, we do see a dose response effect. So there's a lot of variability in the extent to which people adhere to the intervention. We can use an instrumental variables approach we essentially instrument that variation with treatment condition, and we can back out a sort of decontaminated treatment effect. It's about twice the size of the effect that we observe. So it's like 2.5 to 3% slowing in the rate of aging in the intent to treat, and it's about 5 or 6% in the IV. That in turn corresponds to about a 15 percentage point reduction in mortality risk. If we use the effect size estimates that we pull out of the Framingham Heart Study. So it's sort of cheating, but it's the best that we can do at the moment. And that's equivalent to the effect of a smoking cessation intervention. So not a trivial effect of caloric restriction, although a damn lot of work for the participants. But that leaves us with the question of why this thing proved more sensitive to this intervention. Random chance? Certainly possible. Sample composition has been proposed. We trained the Dunedin-Pace algorithm on a phenotype developed in a cohort aging from 26 to 45. The mean age of the calorie trial participants at enrollment was 38 years with an upper bound of 51. So whereas grim age was trained in older Framingham Heart Study participants, so maybe it was about how old the training data were. The explanation we advanced was it's because we're using a speedometer instead of an odometer. And the sort of metaphor I'd offer you is if you imagine a car speeding down the road, you tap the brakes. The odometer sort of doesn't register that at all. But the speedometer is going to show you something happened. And so perhaps by building a rate measure, we've devised a more sensitive index. And I'm going to show you some evidence that that's the case. So, when we published this paper, we could just sort of wave our hands because there were no other pace of aging clocks, no other molecular speedometers to run the test with, and no other randomized control trials to compare it to. So we went back to data where we had longitudinal observations of biomarkers. These are the Framingham Heart Study offspring cohort. We work in a number of cohorts. I'm just showing you a lot of Framingham. And they have a very different design in the Dunedin study, different biomarkers measured at different points in time, and a heterogeneous age distribution. So at the time of measurement, there's about a 30-year span in age at baseline. So we had to adapt our methods a little bit. But roughly, this is what the data look like if we do that same kind of scaling that we did with the Dunedin study data. And so you can see change in the biomarker panel across the 8 waves of follow-up over about 30 years, as the average subject ages from 35 to about 68. And things increase. We model the pace of aging in a slightly different way in this study. We use a hierarchical model, so we're modeling all 17 of the slopes at the same time. Each slope is a composition of a shared component, a participant-specific component, and a biomarker-specific component. So we're taking what Will called theta, the individual level slope that's shared across the biomarkers. That forms our pace of aging. And because we're following people over a period of the life course, when trajectories of these biomarkers are nonlinear, we're allowing the slopes to vary with a quadratic term, but we're focused on our pace of aging only on the linear component of the slope. So this is sort of the initial rate of change that's estimated in the model, and that's what we model from the methylome. This is just a kind of quick validation that this thing that we built in the Framingham Heart Study to predict, differences between people in aging rate corresponds to their risk of death subsequent to the measurement. So this is actually not, this doesn't account for age differences between the people. So this gives you a sort of doubling of the hazard ratio for standard deviation of pace of aging. If you account for age, that hazard ratio drops to 1.7. Okay, so what do we do then? We model it from the methylome in precisely the same way we modeled pace of aging from it. We take DNA from the last wave of assessment, we extract, we assay methylation, we run the elastic net regression on the composite rate of change phenotype. In this case, we were attempting to follow precisely Steve's procedure in developing GrimAge, because that's an important comparison in the Dunedin study. And to account for age differences in his participants, because he knew that the age was tractable in the methylome, but not informative about mortality risk, he included chronological age as an unpenalized regressor in his machine learning analysis, and also the participant's sex. So these are just sort of normal variables thrown in there with the methylation, so that the methylation algorithm is in GrimAge and here in this Framingham Pace algorithm, picking up chronological age independent mortality risk information. And we can do an okay job of fitting the phenotype. This didn't trouble us too much because Dunedin Pace also fits the phenotype about this well. And what we see in the Dunedin study as well as in the Framingham Heart study is that this imperfect proxy delivers essentially the same effect sizes as the original longitudinal phenotype, suggesting that what's being recovered is signal and what's being lost is noise. Okay, so anyway, that's sort of long and technical, but we also we did the same thing and left out the age and sex as unpenalized covariates, just so we had sort of a pure methylation only version of the algorithm as well. And it sort of gives us essentially the same quality of prediction. They are predictive of mortality. So these are data from the Women's Health Initiative and the Health and Retirement Study. Here we're looking at, I think, 18 years of follow-up, and here we're looking at, this is only 6 years of follow-up. And you can see the effect size ranges are a little different, but the Framingham Pace algorithms are on the left side. They're next to Dunedin Pace, and then you have GrimAge and PhenoAge. And you can see, like, again, we're delivering comparable precision in prediction of survival to the other metrics. This is what the data look. So that was a lot of setup for this one slide, but I wanted to take you there. So this is the Dunedin Pace result. These are the Framingham Pace results. This is with age and sex as unpenalized regressors, so as part of the algorithm. This is pure DNA methylation. And then here are the PhenoAge and GrimAge results. And these are the effect sizes of the intervention at 12 months is the top bar, and then 24 months is the bottom bar. So pretty consistently, we see that even when we use exactly the same training data as one of the second generation survival prediction clocks, we can discover an algorithm that is comparably predictive of I just showed you lifespan data, but we have data that suggests health span is also included in that statement. And which is more sensitive to the effects of intervention. Okay. So we have until the end of the hour, right?
Well, but we leave at least.
Yes, I'm gonna give you 15 minutes for questions. I'm just gonna cover very briefly a couple of cute examples of how we've deployed these in somewhat amateurish versions of the work that you guys do. So Gloria Graf was a PhD student working with me. These are two of her papers deploying these indices of biological aging, including our pace of aging epigenetic clocks, in sort of classical health disparities study designs. So here we were interested in how algorithms that had been devised in almost entirely white cohorts would perform as indices of health disparities. And so what we wanted to do is make sure first that they predicted outcomes comparably across groups. And here we're using Black and white Americans in the HRS. And we saw that they in fact did deliver comparable predictions of those outcomes. And then we test mediation, like can we explain the disparity in health span and lifespan by these indices of biological aging. And what we find across a sort of a range of different blood chemistry and DNA methylation indices is you can explain between 20 and 80% of Black-white disparities in a wide range of phenotypes. So I tend to say about half appears to be measurable from this biological aging phenotype in the HRS. The other papers were essentially about establishing that there was something we could do about disparities in biological aging. One of the observations included in our initial paper was that I'm going to show it to you in a minute, actually, but it shows that childhood socioeconomic differences correspond to differences in biological aging. These papers that Gloria was writing were about social mobility. So if we look at educational mobility in Framingham or broader indices of social and economic mobility in the HRS, we see that upwardly mobile people tend to be aging more slowly than people who are stably disadvantaged. This is the childhood SES result. So these are actually 18-year-olds. So we can see social gradients in the pace of aging epigenetic clock in particular. The others are maybe less sensitive in childhood. It's not clear. This is family SES, and these effects can be interpreted sort of on a Cohen's d scale. So these are standard deviation units. It's an effect size about 0.4 from kids who grew up poor to kids who grew up wealthier. And this is blood sampled at age 18. And this is childhood victimization, an even larger effect size. So they're sensitive to things that we think might drive differences between people in aging. Laurel Raffington, now at Max Planck, has done some work in even younger kids using saliva DNA, again, reproducing this observation of a social gradient in health. In Laurel's work, again, it tends to be the speedometers that are more sensitive in the kids. But we just don't have enough data to make any kind of strong statement about that. This was a study. We were really excited. We were going to have the first natural experiment study of an epigenetic clock, and we had this perinatal insult identified by this incredible cohort study that my colleague Bertie LeMay and with molecular expertise from Bas Heijmans had developed. The Dutch famine was caused by a Nazi blockade of the southern Netherlands at the end of World War II, caused a flash famine. It's really a scar on the Dutch psyche, although the biodemographers are now saying it was like not that big of a deal, mortality-wise. Anyway, there's been a lot of research on the children who survived that famine, in particular who were in utero at the time, as a sort of quasi-experimental test of the Barker hypothesis, which proposes that in utero undernutrition conditions risk for a wide range of cardiometabolic disease later in life. And so what I'm showing you here is just the design of the study. It includes babies who are exposed to famine at different points in gestation, and then controls born in the same hospitals before or after the famine. And not shown here are actually some siblings of these folks. Dalton was part of the grant that gave rise to this study, and contributed, in fact, to the journal. So this is the result. We pool the data between the men and the women. We see that 60 years after exposure to prenatal famine, the exposed babies are aging faster than the unexposed babies. Although surprisingly, to me at least initially, this effect is specific to men, or sorry, to women. That was, I think, I was expecting the others. So we can talk about why that is. We have some ideas. But in this cohort, the effect is restricted to women. I don't really have time to go into detail on this work, but I know many of you work with the HRS. And Nature Aging will publish later this spring a pace of aging phenotype in the HRS that we've developed. And eventually the data will be available from HRS, but if any of you want the variable, you can write to us and we will send it to you. It's also available in the English longitudinal study of aging. So here we're putting together data on some blood chemistry analytes, blood pressure, lung function, anthropometry, grip strength, balance, and gait speed. And one of the interesting observations here, we see this with Framingham Pace too, is sort of replication of the demographic observation that the force of mortality accelerates at advancing ages in the biomarker data. So what we see is the rate of aging is also accelerating as people grow older. These are some validation data. Okay, so just here, I'm going to end with the idea that geroscience and its tools open up new frontiers for population health research. There are lots of things that can be done with these measurements of biological aging, risk stratification, population surveillance ultimately is a place where I'd love to see this better integrated into population science. Gloria has a dissertation that is attempting that. But ultimately, the most immediate application, I think, is for the evaluation of policies and programs. This is the sort of caveat that, like, we can show these things predict mortality, we can show they may be modified by interventions, but until we have all the data to establish that they mediate the interventional effect on mortality, we can't really be certain in these tools. I showed you data from a caloric restriction trial. In another year or two, we should have data from these two social policy trials. With Peter Munig, we are collecting data from participants in an employment intervention called My Goals for Employment Success to find out whether a 3-year policy cash and kind of coaching intervention designed to help people get and maintain jobs slowed their rate of aging 3 years after the conclusion of the intervention. And with Allison Aiello and Ashu Honda at Carolina, Allison's now at Columbia, we are generating data from the Malawi Social Cash Transfer Program trial that Ashu convinced the Malawian government to engineer, where they randomized villages to early enrollment or waitlist controls in a national cash transfer program. And so we're going to be comparing 6 years of transfers to 3 years of transfers, basically when the data come through. And then, finally, now that ARPA-H is sort of in limbo, it's unclear if this is really going to live the way it's supposed to. But with Nir Barzilai and some help from Madi Mokri at Harvard, we are trying to pull blood samples out of the freezer from completed clinical trials of drugs that Nir and his team have identified as showing geroprotective potential in animals. So these are drugs that, when you give them to mice, modify hallmarks of aging, and that when we look at clinical observational data, appear to be associated with reductions in all-cause mortality. You've all heard about GLP-1s now. This is Ozempic. It's good for everything apparently, except for muscle. So you have to eat like a whole animal every day and lift a ton of weights, or you will atrophy. Preserved HF is an SGLT2 trial. This is a new diabetes drug. Metformin is an old diabetes drug with a lot of evidence for geroprotective potential, but weak effects. And bisphosphonates are the ones that I was not familiar with beforehand. These are drugs that we give people to prevent bone loss with aging, but they show some promise. So we have actually SGLT2 data just back from SomaScan. So we're gonna have an answer on one of these soon, and then hopefully open them. All right. You can reach us at Columbia. We can help you do this stuff. This is our software. Thank you to all of my collaborators and funders. So, and now I have actually only 10